My name is Caitlin Fisher-Reed. I'm a professor here in the biology department and I teach several courses. Every fall I teach ecology, which is a sophomore level core class that focuses on how organisms live and interact with their environments. Every spring I teach evolution, which is an upper level elective that focuses on evolutionary concepts like natural selection, genetic drift, and the origin of species. I also teach several elective classes that are in my fields of interest. So Darwinian medicine, we discuss all of the ways that evolutionary concepts can be applied to medicine. Then in behavioral ecology, we try to understand why animals do what they do. And in molecular ecology, we use the tools of genetics to try and answer ecological and evolutionary questions. My research interests are really diverse. I take advantage of both fieldwork and lab work. I'm an evolutionary biologist and a molecular ecologist. And that means that I'm combining genetic and habitat data to try and answer ecological and evolutionary questions. I have two main projects right now. I have a long-term monitoring project on salamanders, and then I have a milkweed common garden experiment. So for the salamanders, I'm really interested in how populations of the eastern redback salamander are ecologically and genetically different from each other, and if those differences correspond to differences in their habitat. This work has implications for how new species form, how salamanders and other species are responding to climate change, and just general understanding of populations. For the milkweed, I'm working in collaboration with Dr. Heather Morella here on campus, and we've planted 40 milkweed plants that originate from all over the Northeast. We're keeping track of their growth and reproduction over the next several years to see if any of the locations show particularly strong or weak growth in our local climate. My research would not be possible without a dedicated group of undergraduate research students. We get together every Friday to work on our various projects, and then I usually meet with students one-on-one during the week. When students first join my lab, they spend the first semester or two learning the ropes on the milkweed project and the salamander project. They work closely with me during this time, but also with more senior students on their independent projects to get a feel for what an independent project might be like. After their first semester, I meet with students to talk about what they would want to work on independently, and students in my lab that do have shared interests will often work together to complete these projects, and then all students will go on to present their work at regional conferences in our field. In the lab, we primarily do DNA extraction and PCR, and so we extract DNA from salamander tail clips, milkweed roots, and soil. And then PCR is polymerase chain reaction. We use that to amplify the genes that we're interested in. And then we send those amplifications out for sequencing, and we compare the sequences that we get back to each other. In the field, we use remote data loggers to continually measure temperature, humidity, and light levels. We also use camera traps to monitor larger animals in the forest, and we use pine boards as standardized artificial cover for salamanders. When we catch the salamanders, we measure their size using calipers, and we mark them with an injectable fluorescent compound to give them a unique color pattern that lasts through their lifetime. For milkweed, we also use calipers and measuring tapes to do most of our measurements. Undergrad research is really important to help students actually do science. In the classroom, it's really easy to think that scientific knowledge is a done deal. We know how everything works and what to expect when we test particular hypotheses. That's why it's in the textbook, it's why it's in the lecture. But the reality is that's not the case at all. Science is all about the unknown, asking questions, forming hypotheses, testing those hypotheses, and then making sense out of your results. Even if your experiment rejects your hypothesis, that rejection is still knowledge we didn't have before you did the experiment. So everything you do in undergrad research, no matter whose lab you're in, will be relevant to a career in science, because at the core, we're all doing the same thing, using the scientific method to understand the world around us, whether we focus on molecules within the human body or salamanders out in the woods. So if you're interested in learning more about me, the research I do, or the classes I teach, please visit my website. Thank you for listening, and I hope to see you in class.